Pyogenic infections are infections that lead to the formation of pus. In Theme 3.6, we will look at gram-positive rods that cause pyogenic infections. They can be divided into two families, the Corynebacteria Actinomyces and the Actinomyces Tpyogenes. Diseases caused by the Corynebacteria Actinomyces are caseous lymphadenitis of sheep and goats, pyelonephritis of cattle, and Bola disease. Then we will look at infections caused by Actinomyces bovis and Tuberella pyogenes, members of the Actinomyces Tpyogenes family. The Corynebacteria Actinomyces are small gram-positive club-shaped rods that are primarily aerobic and facultatively intracellular. They are widely distributed in nature, and many are non-pathogenic. The pathogenic species include animal and human pathogens. These bacteria are often associated with abscesses. The first disease on our list is caseous lymphadenitis, also known as CLA. It is a chronic and subclinical disease of sheep and goats that occurs worldwide. It presents with high prevalences in animals and flocks. The disease is caused by Corynebacterium pseudotuberculosis. This is not a controlled disease, even though it causes economic losses due to carcass condemnation. Carrier animals are the main reservoir for this infection. The environment is contaminated by infected active carrier animals. When abscesses rupture, infection occurs via damaged skin or mucous membranes. Other routes of infections are inhalation of aerosols from animals with lung infections or ingestion of contaminated feed or water. At the point of entry, the bacteria are phagocytized by neutrophils or macrophages. The bacteria produce exotoxins that protect them against lysosomes in these cells and cause increased vascular permeability. From the initial infection site, the bacteria are moved to the local lymph nodes. From here, they can disseminate to any organ in the body, as well as the joints, and cause abscesses. High levels of environmental contamination is often the source of infection. Corynebacteria pseudotuberculosis can survive in organic materials and nutrient-rich soil for months or years under cool and moist conditions. Predisposing factors are anything that compromises the body's natural defenses. For example, skin trauma due to shearing, tail docking, castration, or fighting, skin maceration due to constant wet conditions, contaminated needles or instruments, having carrier animals with lung abscesses in the herd, and then crowding the animals post-shearing can lead to aerosol spread. Crowding can also lead to direct contact between open wounds and ruptured abscesses. Corynebacterium pseudotuberculosis can persist in dip for up to 6 months. If animals are dipped directly after shearing, skin wounds can be infected. However, animals are not more susceptible with increasing age. The risk of exposure just increases. Incubation period can be anything from 1 week to 5 months. Affected lymph nodes enlarge and eventually rupture to discharge a thick yellowish-white pus. Animals lose weight and become progressively weaker. This could also be an indication of abscesses in internal organs. According to statistics, sheep are more prone to internal abscesses and goats to external abscesses. Note the predilection sites, where abscesses are most often located: around the head and shoulders, on the hind leg, and in addition between the hind legs where the scrotum or udder attaches. These bacteria grow readily during routine aerobic culture, so a sample can be collected from the edge of the abscess for submission to a bacteriology laboratory. Serological assays exist in the form of an ELISA, but they are not useful in vaccinated flocks. Here are some images of abscesses. These lesions can inspissate and appear very similar to Mycobacterium bovis lesions. The most effective control of this disease is achieved by a combination of vaccination, clinical examination, and culling of affected animals in combination with appropriate hygiene measures. In South Africa, an inactivated vaccine is available under the old name for this organism, Corynebacterium ovis. The immune response to vaccination only lasts 2 to 3 weeks, which necessitates strategic use. Only in affected flocks and only before an event such as shearing or castration. In other countries, live vaccines are also available, but they cause unacceptable local reactions at the injection site. Vaccination on its own can also eliminate the infection from a herd, but only if the prevalence is below 60%. Other options. Lancing is not advisable because it does not address internal carriage. Such animals could serve as carriers and reservoirs. Furthermore, the environment is likely to be infected by the bacteria in the pus. Clinical examination and culling is effective, but progress may be slow because animals with internal abscesses are not identified. Serological testing and culling can only be used in unvaccinated herds. This method can only be used if the sensitivity and specificity of the test is more than 90%. Now for more members of the Corynebacterium genus. This time, I will present a case study to introduce them. 3 adult Guernsey cows in a dairy herd present with the following clinical signs: decreased milk production, loss of condition, varying anorexia, decreased rumen motility, a tense abdominal wall, and frequent and painful urination in 2 of the cows. Additional history is as follows. All the cows are on a high protein diet. They are on a zero grazing system. They are high milk producers in their 5th or 6th lactation, and 2 of the 3 had given birth less than 3 months before. The clinical examination includes collection of midstream urine due to the signs of frequent urination noted. Urine collected from the 3 cows appear as follows. One is bloody, another has a sediment that appears to be red blood cells. All are cloudy and they have an offensive odor. Urinalysis detects proteins, blood and leukocytes in the urine, an indication of an infection. On a rectal examination, dilation of the left ureter is detected in 2 cows, and enlargement of the left kidney is detected in 1 cow. In the laboratory, further workup of the urine sample reveals a large number of small club-shaped bacterial rods, pointing to a bacterial infection. Blood hematology and biochemistry results include high creatinine levels indicating kidney disease or impaired kidney function, as well as high urea levels also indicating impaired kidney function. Accumulation of fluid in the body leads to a dilution of the sodium and can be caused by kidney disease as well. All the results of the clinical and laboratory investigation point toward a bacterial pyelonephritis. Corynebacterium renale is cultured from urine samples in all three cases. In 2 cases, the culture is mixed with Streptococcus agalactiae. Streptococcus agalactiae is a commensal of mucous membranes that will cause secondary infections in the presence of a primary infectious agent, as in this case. An antibiogram indicates that the bacteria are susceptible to penicillins, tetracyclines, cephalosporins, and macrolides. An alternative to culture is sometimes a serological test. An IFA, indirect fluorescent antibody test, exists by which Corynebacterium renale kidney infection can also be confirmed. However, if this route is taken, an antibiogram is not available because a live culture is required. So, there are 3 species of Corynebacterium that can cause the disease, contagious pyelonephritis. These are Corynebacterium renale, the most infectious, Corynebacterium cystitis, the most virulent, which usually only causes cystitis, and Corynebacterium pilosum, the most benign. These bacteria exist as commensals in the external urogenital tract of cattle and sheep. Most of the causative bacteria of this disease are commensals of the vagina and prepuce. Pyelonephritis develops due to an ascending infection from the bladder. The causative bacteria will colonize the bladder and ureters following an incident that causes trauma, for example parturition, or due to deformity of the vaginal tract. A high-protein diet together with parturition stress lead to an increased pH, which facilitates growth of bacteria. Other risk factors are urine aerosol, frequent or careless use of AI catheters, age. Multiparous cows are more likely to develop disease, and build-up of organisms in the environment due to poor hygiene. Cows with advanced kidney damage, indicated by high creatinine levels, should be culled due to poor prognosis. Mildly affected cattle may be treated with penicillin or with other gram-positive spectrum antibiotics, according to antibiogram results. A urine acidifier can be given. Fluids can be given to maintain renal perfusion. The images are of the inside of a bladder with a congested bladder wall and a plaque. At the bottom, the kidney is swollen. Note the loss of the lobular structure. To prevent further spread, and bearing in mind that affected cows will shed large numbers of bacteria in the urine, known positive cows should be separated and treated. Milk these cows last, and discard the milk until after the withdrawal time of the antibiotic. Keep new cattle separately. Bola disease is a unique disease caused by a Corynebacterium species. It was named after the farm Bola, where it was first diagnosed in 1974. Infection by these Corynebacteria causes a superficial dermatitis in wild sheep. It occurs in parts of KwaZulu-Natal and the Eastern Cape. The disease causes losses due to downgrading of fleeces and higher labor costs, because affected wool has to be plucked from shorn fleeces by hand. This is a chronic infection that results in long-term production losses. The infection is usually mild and localized. Lesions appear after an incubation period of 2 to 9 months. These lesions are circumscribed patches where the wool is darker and shorter. Actinomycosis or lumpy jaw is characterized by a focal pyogranulomatous osteomyelitis, resulting in bone necrosis followed by bone proliferation. The mandible is affected most often. Actinomyces bovis is a small gram-positive rod-shaped bacteria. It is a commensal of the oropharynx. Colonization takes place early in the animal's life by transfer from the dam, so any form of oral trauma can lead to submucosal infection. A few images of characteristic lesions. Actinomyces bovis can reach submucosal tissue or the periodontal membrane following mucosal trauma due to eruption of teeth, coarse feed or foreign bodies. It infects and multiplies, leading to a necrogranulomatous inflammation. The infection spreads to the underlying bone, leading to a pyogranulomatous osteomyelitis. The result is bone necrosis and proliferation of granulation tissue, with the formation of abscesses and sinus tracts. The content eventually becomes mineralized, presenting as sulfur granules, which is characteristic of this infection and which can be seen in the pus. This infection leads to the enlarged honeycomb appearance of the jaw, which led to the name lumpy jaw. Further complications are teeth malalignment and loosening, fistulas, and pathological fractures. Individual animals are usually affected. Lesions develop chronically over months. The swelling is bony, fixed, and appears painless. Sinus tracts may be present. This organism can also cause fistulous withers in horses. Malalignment of teeth is a painful complication of this infection and affects the animal's ability to feed, resulting in emaciation. Pathological jaw fractures can occur due to the weakened state of the bone. The infection can spread to soft tissues as well. In rare cases, the infection can spread to the regional lymph nodes, lungs, liver, and vertebra. Trupperella pyogenes infections. Trupperella pyogenes, formerly known as Corynebacteria pyogenes, is a small gram-positive rod that forms very small white colonies with beta hemolysis on blood agar. This bacteria had many names over the years. These names may be encountered in literature and on vaccine labels. Trupperella pyogenes is a commensal of the mucosa of the upper respiratory tract and genitals. It is an opportunistic pyogenic bacterium. It is the most common pyogenic infection in cattle. It usually causes a grayish to yellow-green malodorous pus. Similar to Actinomyces bovis, Tpyogenes Pyogenes requires some form of damage to the mucosa to gain access to the submucosa and cause infection. Damage can be caused by trauma or viral or other bacterial infections. The bacteria can enter and survive in epithelial cells and macrophages. It can cause a wide range of suppurative infections, also mixed infections with anaerobes. The word suppurative indicates the nature of a lesion or inflammation that is forming pus, or the act of becoming converted into and discharging pus. These are some images of the most common infections due to Tpyogenes pyogenes: liver abscesses, pneumonia, foot rot, and mastitis. The role of Tpyogenes pyogenes in mastitis is that of an environmental pathogen introduced, for example, following bites of ticks with long mouthparts. Ruptured abscesses can lead to toxemia and even death. It can also be involved in mixed pyogenic mastitis. T-tips are often entry points following insect bites. Tpyogenes pyogenes can act synergistically with anaerobes. Tpyogenes pyogenes uses oxygen, so it lowers the oxygen levels in tissues, which make it a more suitable environment for anaerobes. Anaerobes, such as Bacteroides species, can protect Tpyogenes pyogenes from neutrophil phagocytosis. Tpyogenes pyogenes also produces a factor that enhances the growth of Fusobacterium necrophorum, while Fusobacterium necrophorum produces leukotoxin that protects both organisms from phagocytosis. In the case of Tpyogenes pyogenes abscesses can be excised and managed as open wounds. Antimicrobial therapy alone has limited success because the bacteria may be intracellular and abscesses are encapsulated. Tpyogenes pyogenes are usually sensitive to any gram-positive spectrum antibiotic, for example, beta-lactam antibiotics, macrolides, tetracyclines, and sulfonamides. In case of a mixed infection with anaerobes, metronidazole can also be considered. Avoidance of traumatic skin injuries is an important part of management where these bacteria are a problem. So tick control and avoidance of pastures, coarse or thorny feed as well as coarse bedding is essential. Dosing must be performed carefully and dosing guns must be free of burrs. Strict hygiene must be practiced during traumatic procedures and in areas where animals give birth. A vaccine is available in the form of an alum-precipitated bacterium and toxoid. Note that it is marketed under two of the older names for these bacteria. The vaccine for Tpyogenes pyogenes should only be used on problem farms and must be used in conjunction with good on-farm hygiene. It is administered every 6 months from 6 months of age.